membranes, their fibers become continuous with those of the areolar tissue. The sarcolemma, or tubular sheath of the fiber, is a transparent, elastic, and apparently homogeneous membrane of considerable toughness, so that it sometimes remains entire when the included substance is ruptured. On the internal surface of the sarcolemma in Mammalia, and also in the substance of the fiber in frogs, elongated nuclei are seen, and in connection with these is a little granular protoplasm. Upon examination of a voluntary muscle fiber by transmitted light, it is found to be marked by alternate light and dark bands or striae, which pass transversely across the fiber. When examined by polarized light, the dark bands are found to be doubly refracting, anisotropic, while the clear stripes are single refracting, isotropic. The dark and light bands are of nearly equal breadth and alternate with great regularity. They vary in breadth from about 1 to 2 microns. If the surface be carefully focused, rows of granules will be detected at the points of junction of the dark and light bands, and very fine longitudinal lines may be seen running through the dark bands and joining these granules together. By treating the specimen with certain reagents, for example chloride of gold, fine lines may be seen running transversely between the granules and uniting them together. This appearance is believed to be due to a reticulum or network of interstitial substance lying between the contractile portions of the muscle. The longitudinal striation gives the fiber the appearance of being made up of a bundle of fibers which have been termed sarcostyles or muscle columns, and if the fiber be hardened in alcohol, it can be broken up longitudinally and the sarcostyles separated from each other. The reticulum, with its longitudinal and transverse meshes, is called sarcoplasm. In a transverse section, the muscle fiber is seen to be divided into a number of areas called the areas of Cohnheim, more or less polyhedral in shape and consisting of the transversely divided sarcostyles surrounded by transparent sarcoplasm. Upon closer examination, and by somewhat altering the focus, the appearances become more complicated and are susceptible of various interpretations. The transverse striation, which appears as a mere alternating of dark and light bands, is resolved into the appearance which shows a series of broad dark bands separated by light bands, each of which is divided into 2 by a dark dotted line. This line is termed Dobbe's line or Krauss's membrane because it was believed by Krauss to be an actual membrane continuous with the sarcolemma and dividing the light band into 2 compartments. In addition to the membrane of Krauss, fine clear lines may be made out with a sufficiently high power crossing the center of the dark band. These are known as the lines of Henson. Schaefer has worked out the minute anatomy of muscle fiber, particularly in the wing muscle of insects, which are peculiarly adapted for this purpose on account of the large amount of interstitial sarcoplasm which separates the sarcostyles. In the following description, that given by Schaefer will be closely followed. A sarcostyle may be said to be made up of succession portions, each of which is termed a sarcomere. The sarcomere is situated between 2 membranes of Krauss and consists of 1. A central dark part, which forms a portion of the dark band of the whole fiber and is named a sarcous element. This sarcous element really consists of 2 parts, superimposed one on top of the other, and when the fiber is stretched, these 2 parts become separated from each other at the line of Henson. 2. On either side of this central dark portion is a clear layer, most visible when the fiber is extended. This is situated between the dark center and the membrane of Krauss, and when the sarcomeres are joined together to form the sarcostyle, constitutes the light band of the striated muscle fiber. When the sarcostyle is extended, the clear intervals are well marked and plainly to be seen. When, on the other hand, the sarcostyle is contracted, that is to say when the muscle is in the state of contraction, these clear portions are very small, or they may have disappeared altogether. When the sarcostyle is stretched to its full extent, not only is the clear portion well marked, but the dark portion, the sarcous element, is separated into its 2 constituents along the line of Henson. The sarcous element does not lie free in the sarcomere, for when the sarcostyle is stretched so as to render the clear portion visible, very fine lines, which are probably septa, may be seen running through it from the sarcous element to the membrane of Krauss. Schaefer explains these phenomena in the following way. He considers that each Sarcus element is made up of a number of longitudinal channels, which open into the clear part towards the membrane of Krauss, but are closed at the line of Henson. When the muscular fiber is contracted, the clear part of the muscular substance is driven into these channels or tubes, and is therefore hidden from sight. But at the same time, it swells up the Sarcus element and widens and shortens the sarcomere. When, on the other hand, the fiber is extended, this clear substance is driven out of the tubes and collects between the Sarcus elements and the membrane of Krauss, and gives the appearance of the light part between these two structures. By this means, it elongates and narrows the sarcomere. If this view be true, it is a matter of great interest and, as Schaefer has shown, harmonizes the contraction of muscle with the amoeboid action of protoplasm. In an amoeboid cell, there is a framework of spongioplasm which stains with hematoxylin and similar reagents, enclosing in its meshes a clear substance, hyaloplasm, which will not stain with these reagents. Under stimulation, the hyaloplasm passes into the pores of the spongioplasm. Without stimulation, it tends to pass out, as in the formation of pseudopodia. In muscle, there is the same thing, that is, a framework of spongioplasm staining with hematoxylin, the substance of the Sarcus element, and this encloses a clear hyaloplasm, the clear substance of the sarcomere, which resists staining with this reagent. During contraction of the muscle, that is stimulation, this clear substance passes into the pores of the spongioplasm, while during extension of the muscle, that is when there's no stimulation, it tends to pass out of the spongioplasm. In this way, the contraction is brought about. Under stimulation, the protoplasmic material, the clear substance of the sarcomere, recedes into the Sarcus elements, causing the sarcomere to widen out and shorten. The contraction of the muscle is merely the sum total of this widening out and shortening of these bodies. Vessels and nerves of striped muscle. The capillaries of striped muscles are very abundant and form a sort of rectangular network, the branches of which run longitudinally in the endomysium between the muscle fibers, and are joined at short intervals by transverse anastomosing branches. In the red muscles of the rabbit, dilatations occur on the transverse branches of the capillary network. The larger vascular channels, arteries and veins, are found only in the perimysium, between the muscular fasciculi. Nerves are profusely distributed to striped muscle. Their mode of termination is described on page 730. The existence of lymphatic vessels in striped muscle has not been ascertained, though they have been found in tendons and in the sheaths of muscles. Ossification of muscular tissue as a result of repeated strain or injury is not infrequent. It is oftenest found about the tendon of the adductor longus and vastus medialis in horsemen, or in the pectoralis major and deltoideus of soldiers. It may take the form of exostoses firmly fixed to the bone, for example, quote, rider's bone, end quote, on the femur, or of layers or spicules of bone lying in the muscles or their fascia and tendons. Bucy states that these bony deposits are preceded by a hemorrhagic myositis due to injury, the effused blood organizing and being finally converted into bone. In the rarer disease, progressive myositis ossificans, there is an unexplained tendency for practically any of the voluntary muscles to become converted into solid and brittle bony masses, which are completely rigid. End of section 26. Recording by M.L. Cohen, Cleveland, Ohio, www.frontalcortex.com.